This conference will now be recorded.
Good evening, students. I'm Dr. Divya. So now we are in the fifth chapter, Principles of Inheritance and Variation. So this explains about the laws and rules which governs the inheritance of a character and the variations which is created through each generation. So each generation will be having some kind of variation compared to the parental generation, right? So this chapter, Principles of Inheritance and Variation, this explains the complete inheritance and variation of each generation. Okay, what are all variations or what are all characters we will get inherited from our parents and how it is getting inherited, how variations are created when a character is getting inherited from a parent. So in this chapter, throughout, we can learn all these things. And in the previous classes, we learned about genetics. That is a branch of biology which deals with the principles of inheritance and the variation, all those practices. Okay, so progeny, that is resembling the parents in morphological and physiological features. And that attracted the attention of many biologists. And Mendel was the first person who studied this phenomenon in a systematic way. And while studying the pattern of that inheritance in pea plants, which is having different contrasting characters, Mendel, throughout his study, he maintained a good data, collection of good data, and Mendel proposed the principles of inheritance. And these are today referred as Mendel's law of inheritance, right? We are referring these laws as Mendel's law of inheritance, and about the inheritance pattern he is explaining in this laws. So he proposed that the factors of the genes, now we are calling they are factors as genes. So these genes are factors, they are regulating the characters, and these are existing in pairs. These pairs are called as alleles. Okay, this factor, they are existing as pairs, and these pairs are called as alleles. And he observed that the expression of the character in the offspring, which is in a particular pattern, from the first generation to second generation, it is flowing in a pattern. So there is a pattern in this inheritance and the creation of variation. Okay. So some characters are dominant and some characters are recessive. The expressing character, that is called as dominant character, and the hiding or the masking character, that is called as recessive. So the dominant characters are expressed when factors are in heterozygous condition. Definitely in homozygous condition, only one character, one allele is present, so definitely it will be expressing. But when it is in heterozygous condition, the dominant allele is present, that character will be present or that character will be expressed. This is the law of dominance. Got? From the name itself, from the term itself, it's clear. Law of dominance, how one character is getting that dominant effect. When the dominant allele is present, definitely that will be expressed. That is the law of dominance. Then the recessive characters, they are only expressed in homozygous condition. Okay. When it is in homozygous condition, only in that condition, the recessive characters will be expressed. And this character will never blend in heterozygous condition because already there is a law of dominance. When the character or the genotype, it is in heterozygous condition, only the dominant allele will get expressed. That is the law of dominance. Then how that recessive character will get expressed, or when that recessive allele will get a chance, when it is coming in a homozygous condition, only in that condition the recessive character will be expressed. Okay. So character that was not expressed in heterozygous condition, that may express again when it comes to homozygous condition. Okay. When the law of dominance, according to law of dominance, only dominant character will be expressed. Right. So when it comes to the recessive, when that recessive character will get a chance, when it is in a homozygous recessive condition. But when the F2 generation comes, in F1 generation, we have already told, in F1 generation, only one parental character will get a chance to get expressed, right? Because of the law of dominance, the dominant allele presence will be there. So the dominant character will get expressed. So there is only one parental phenotypic expression. When it is coming to F2 generation, here in this picture itself, you can see when it is coming to F2 generation, both the parental characters will get a chance to express. Okay. So the characters are getting segregated through the gametes. In F2 generation, both the characters will get a chance to get expressed. That is the law of segregation. Okay. So there are two laws which we have already learned: law of dominance and law of segregation. Okay. So last class we discussed about codominance, right? Codominance. What is that? In this, we can see that the F1 won't be showing any resemblance with both the parents. Okay, codominance means there is both dominant alleles, both alleles will be they will work in a coordinated way. Both will get expressed. Okay, so in this, we can see that F1 won't be having any resemblance with any of the parents. Okay, both the parents will be different from the F2 expression, or the F1 expression will be different from the parental expression. Okay, so according to Mendel's law, F1 resembles either of the two parents, or it will be in an incomplete dominance, or there will be an incomplete or in between expression, right? We have learned about the flower color in Snapdragon. In that, it was showing incomplete dominance, because when red and white color flower crossed, there was a pink color appearance, right? So there was a separate appearance in the F1. There was an incomplete. It was not going towards red color or not towards white color flower. It was an in-between expression, like a pinkish expression that showed. That is the incomplete dominance. But according to Mendel's law, F1 should resemble any of the parent, right? Otherwise, it should be in an incomplete dominance. But in codominance, we learned about codominance in last class. In codominance, what we can see, all these conditions, there was extremely different condition in the case of codominance, right? In that concept, or in the concept of codominance, F1 generation resembles both the parents. Okay, that is the difference. When codominance concept comes, the F1 generation that resembles both the parents. And we have discussed about very good example for this, for codominance. What was that? ABO blood grouping in human. Okay, and based on that, we learned about 6 different genotypes of human ABO blood types. 6 different genotypes. Okay, and we learned about 4 phenotypes. 4 phenotypes like A, B, AB, and O. That was the 4 genotypes we learned, and there were 6 different genotypic expression. Genotypically, 6 different expressions we can see. And ABO blood group of human, that was an excellent example for multiple alleles also. Multiple alleles means more than 2 alleles for a gene. Usually we will say for one gene, 2 different forms, one dominant and one recessive forms, or slightly altered forms. That is called as alleles, right? So usually, in normal cases, there will be 2 alleles, but sometimes there will be multiple, more than 2 alleles, existence of more than 2 alleles for a gene. So that also will give same kind of character, okay? But the alleles will be slightly different. So ABO blood group, it is an example for codominance and multiple alleles. Multiple alleles means there will be 3 different types of alleles, right? IA, IB, and small I, which shows different types of sugars on the surface of RBC. So on the surface of RBC, different types of sugar. Based on that, you will get 3 different types of alleles. That is an example for multiple alleles, and with the same thing we can explain codominance. When IA and IB comes together, we can see AB blood group, existence of AB blood group. That is an example for codominance. Both will show the presence. Okay, both will show the expression. So next we are learning about starch synthesis. Okay, in today's class we are going to learn about starch synthesis in pea seeds. Okay, starch synthesis in pea seeds. That's what we are going to learn. This is the starch synthesis that in pea seeds that is controlled by one gene. Okay, normally one gene is controlling the starch synthesis. Okay, in pea plant we are saying pea plant, one gene is controlling the starch synthesis. Okay, so this one gene is having two alleles. I'm talking about one gene. Okay, one gene is having two different alleles. What are the alleles? Capital B and small b. These are the two different alleles of that starch synthesizing gene. Okay. So listen, this questions, you may get questions from this, so many questions. So you should be clear about the theory part, then only you can solve the application type questions. You'll get a lot of questions for NEET exam as well as for your board exam. You can see so many questions. So careful. So listen properly. So when this dominant B here, this is the dominant allele. Here I'll mark it as dominant. Okay. And this is the recessive allele. So here one gene which is helping for starch synthesis in pea plant or pea seeds, and in this you can see that one gene is having two different alleles, capital B that is the dominant allele and small b that is the recessive allele. So when this dominant allele B, it is coming in homozygous condition, then starch synthesis is effective. Okay. When this, which is the dominant allele? This one. Dominant allele, when it is coming in homozygous condition, means both alleles are coming in similar way, then you can see starch synthesis in a effective way. Okay, starch synthesis will be high in such seeds. Okay, when the dominant allele is coming in homozygous condition, this is the important point. Homozygous condition, starch synthesis is high. But when the homozygous recessive condition comes, like for this allele, when recessive condition, when this recessive condition is getting an expression, or when it is getting a chance to express, when it is coming in homozygous condition only, recessive expression will come, right? So when it is coming in homozygous recessive condition, you can see starch synthesis in low. Okay, that time starch synthesis will be less. When homozygous dominant condition comes, starch synthesis is high. But when homozygous recessive condition comes, starch synthesis is low. So this produces smaller starch grains. When it is in recessive condition, the starch gains, here you can see large starch grain. When it is coming in this condition, you can see smaller starch grains. Okay, smaller starch grains in this one. Okay, and here this capital B, homozygous dominant condition. One more thing is that the seeds are round. You can see round seeds which are having high starch. And when it is coming to recessive condition like small b, you can see wrinkled seeds with low starch. Okay, homozygous recessive condition, wrinkled seed with low starch. Next one chance is that heterozygous condition. Right now we have already discussed about homozygous dominant and homozygous recessive condition. And next chance is what? That is heterozygous dominant condition, right? Like this also one combination is possible. So when it comes in capital B, small b, that is heterozygous condition, definitely here dominant allele is present, right? Capital B, which is the dominant allele, that is present. So that shows the dominance, and they shows intermediate size. Okay. Intermediate size starch grains. So if you are concentrating on the size of starch grains, here also in this condition also starch synthesis is taking place, but it is an intermediate condition. So when we are concentrating on the size of the starch grain as a phenotype, then heterozygous condition that shows incomplete dominance. Got it? Even the presence of this dominant allele. Here, usually, according to Mendel's law, when a dominant allele is present, that will express in dominant condition. So, according to that, when this B, capital B, the round and high starch grain condition, that allele is present here, right? So, according to Mendel's law, that should express round seed with high starch synthesis, right? But here in heterozygous condition, that shows a different expression. That shows round seed, but the starch grain size is intermediate. Here you can see intermediate. So that's why it is called as incomplete dominance. Okay, here the dominance is incomplete. Here the B allele, which is the dominant allele, that is not showing complete dominant character. It is showing incomplete dominance. It's showing dominance. It is synthesizing starch grains, but it is not in the complete form. It's showing some kind of incomplete dominance. So the starch grain size is intermediate. Okay. So one gene in pea plant, one gene is responsible for starch grain synthesis, and that is the B gene in dominant condition and recessive condition. In both conditions, this gene exists. That is having two different alleles, but dominant allele and recessive allele. When it is coming in dominant condition, that helps in the starch synthesis. Okay. That means when the B gene, when it is coming in homozygous condition, homozygous dominant condition, the starch synthesis is high and that starch grains are large. When it is coming in recessive condition, homozygous recessive condition, starch synthesis is low, very low, and the seeds are smaller. Okay, and homozygous dominant condition, that will help in the formation of round seeds, whereas the homozygous recessive condition forms the wrinkled seeds. But when it comes in the heterozygous condition, like capital B, small b, this is another combination, right? Like heterozygous condition, intermediate starch grains. Starch grains are getting synthesized, but they are in intermediate form. Even though the presence of dominant allele is there, then also it is producing, it is not showing complete dominant effect. It is showing an intermediate effect. It is not showing complete dominance. Instead of that, it is showing incomplete dominance. It's dominant, but that dominance is incomplete. It's not forming the whole dominant character. It's incomplete. So it is controlled by the starch synthesis in pea plant or pea seed. It is controlled by one gene and that has two alleles, capital B and small b. So the starch synthesis, starch is synthesized effectively by capital B, That means homozygote. And therefore large starch grains are produced. Okay. So in contrast, small b, that means homozygous recessive condition that has lesser efficiency in starch synthesis and that produce smaller starch grains. So after maturation of the seeds, capital B, that means homozygous dominant seeds, they are round and the homozygous recessive seeds are wrinkled. Okay. So the heterozygote, another condition is heterozygous, capital B, small b, that produces round seeds. Here, as the capital B shows the round seeds, here also it is showing the round seeds. And so B seems to be the dominant allele, but the starch grain, in the case of starch grain, that produces intermediate size in heterozygous seeds. Okay. So if the starch grain size we are considering, the character which you are choosing as starch grain size, then as the phenotype, in the heterozygous phenotype, that shows incomplete dominance. Not in the case of starch grain size, that character we are considering means in heterozygous condition, that is an incomplete, that shows an incomplete dominance. Okay. So next is the inheritance of two genes. Okay. Next is the inheritance of two genes. So here, two gene inheritance. Mendel did his crossing work in pea plant. Okay. So Mendel was monohybrid cross. We have already discussed about monohybrid cross. That there a single character was considered. So next is the concept of inheritance of two genes where Mendel did crossing work in pea plant. He considered two pairs of contrasting characters. Okay. Two contrasting characters. Here two pairs of traits or contrasting characters were considered. So Mendel did crossing work in pea plant that is having two pairs of contrasting character. So here you can see, in this you can see, consider the cross. Here you can consider this cross between pea plants. Here round yellow, round seed shape with yellow seed color. Okay. Here you can see round seed shape with yellow seed color. And the second parent, that was seed shape, was wrinkled. Here you can see the wrinkled seed shape and the seed color, it is green color. Okay, first character was wrinkled and second character was green color seed. Here round shape, yellow color seed. Here wrinkled shape and green color seed. These were the two parents of parental generation which was selected by Mendel. So in the picture you can see parents, round shape, yellow color seed, that with wrinkled shape and green color seed. Okay. So the parents, how we will represent? Round. Round. These are pure lines. Usually we will select pure lines for crossing. So it will be in homozygous condition. So capital R for that round character, we'll mention capital R, and for yellow, again the pure line form capital Y. So this is how we represent the characters. And wrinkle means the recessive form of this round. So smaller, then yellow, that is the, here green, that is the recessive form of yellow color. So small y. Okay, here gametes, you can see the gametes, capital R and small y. These are homozygous conditions. So it will form a gamete which is having round and yellow color allele. Only one type of gamete it will form. All gametes, whichever it is formed, it will be similar, right? Because it is in homozygous condition. In same case, here also, the recessive alleles that will form the gamete, small r and small y. These forms gamete. So all gametes will be having same condition, same genotypic condition, small r and small y. Then this will join to produce Next, F1 generation. In F1 generation, how it will be? Capital R, here, these combination, capital R and small y, then capital Y and small y, right? Here you can see the combination, capital R and small r, capital Y, small y. This forms here for seed shape R, dominant allele R is present. And here wrinkled one, that recessive allele is also present, but the dominant allele is present. So the expression of the seed shape will be round only. That character will be round only. And here the seed color, yellow, allele for yellow and allele for green, both are present, but the dominant allele is present here. So definitely the expression will be for the dominant allele. So round and yellow seeds will be present in F1 generation. Round and yellow seeds will be present in which generation? F1 generation. Okay. So the resulting seeds have round shape and yellow color. Both dominant alleles here for shape, dominant allele is expressed, and for color also dominant allele is expressed. So yellow color was dominant over green color and round shape was dominant over wrinkled one. Okay. So the result of this was also similar like monohybrid cross. From this you can make out the result is similar like monohybrid cross. The dominant allele is expressing, getting expressed. Right. It is exactly like monohybrid result. So here you can see round seeds with yellow seed color that is crossed with wrinkled seed with green seed color. Okay, round means in homozygous condition will express like this and yellow will express like this. Right? And wrinkled, how wrinkled will show? Smaller, smaller. These are the recessive character and green will show like this. So how it will form gamete? Capital R and small y. This is the gamete produced from this. These are the parents and this is the gamete. Okay. And here, which type of gamete it will form? Small r, small y. The recessive type gamete it will form. And this will fuse together here. The fusion will take place and this forms the F1 generation. Here, when two characters are considered, for first character we'll write both the alleles, capital R and small r. Then for yellow color seed, for the seed color we'll write both the alleles. Capital Y, this is small r, here capital Y and small y. So this is the F1 genotype of F1. But here you can see dominant allele is present. There, here dominant allele for seed color, seed shape, dominant allele is present. So definitely it will form round seed. And here for seed color, dominant allele is present, which is yellow. So round and yellow. What will be the phenotypic expression of this round and yellow? Okay. So here, by using symbols, we wrote this. Y for dominant yellow seed color and small y for recessive green seed color. Right? Don't get confused when we are considering two characters. And here R. R for dominant round shaped seed and small r for wrinkle shaped seed. Okay. Then here parents, how the parents will come? For single character that is very easy, it won't be confusing, but here when here two characters are considered, while writing the gametes and all, you should be careful. So the parents will be capital R, capital Y, capital Y, both in homozygous dominant condition. Okay. Then next parent, that is recessive alleles. These are pure lines. That's why we can write very easily. For this round character, we need a recessive gene here. Small r from the other parent and small y. It is for green seed color, which is the recessive character of this yellow seed color. So gametes will be from here one type gamete, capital R, capital Y, and from here small r, small y. These both will combine and it will form capital R, capital Y, small y. Right? These combination. First, which character we are considering? That both alleles we need to write first. Then second character which we have considered, for that also both the alleles which are present in this combination. So here F1, how the expression on fertilization, this will produce F1 generation or F1 hybrid. F1 hybrid will be capital R, small r and capital Y, small y. So what is the phenotypic expression of this? It is round and yellow. Here again, like monohybrid cross, it is giving the one parental expression, right? The phenotypic expression is like one parent, round and yellow. Here, even though it is an heterozygous condition, the expression is round and yellow. Okay. Okay. Next, like monohybrid cross. Same like that. What he did in monohybrid cross, he allowed for selfing. Okay. Like monohybrid cross, Mendel allowed for selfing. And again, with the help of this Punnett square, we can find out all these combinations. So it will be easy. While writing also, this will be easy. The Punnett square. So selfing means how it will be? Selfing means, so now F1 is in capital R, small r, capital Y, small y. This is the genotypic expression of F1, right? Then we are going to do selfing. So how it will form? It will form gametes, right? Selfing means this capital R, capital Y, small y. This is crossing with the same one, capital R, capital Y, small y. So this will produce here 4 different combinations you can see. If it is a homozygous condition, it will produce only one type of gamete. But here it is in a heterozygous condition. So which are all the possible combinations of gametes? Here, first, capital R may combine with capital Y, right? That is one possible gamete. Then this small r, again, this also may combine with capital Y, right? Next, this capital R may combine with capital Y. Sorry, small y. Capital R may combine with small y. Now this combination already over, right? Capital R and small y. Then last, small r with small y. These are the 4 possible gametes from this heterozygous condition. Okay. So in the same way, male gamete and female gamete. Here also these 4 possible combination, and in this side also you can see these same possible combination because it is a self cross, right? It's a selfing. So here we'll see the all possible combinations. Here this capital R, capital Y and capital R, capital Y. The first combination. First we will see the possible combination of this male gamete with all the female gametes. Okay, these are the female gametes. Here we have marked as female and this is the male plant, right? So first we will see the possible combination of this male gamete. First male gamete with all these female gametes. 1, 2, 3, 4. Okay. First, when it is combining with capital R, then another allele. For the same thing, another allele. Small, here also capital R, here also capital R. So these two, first these two will come, capital R, then next character, capital Y, capital Y. Okay, the first combination. The next combination is this male gamete with the second female gamete. The first male gamete with the second female gamete. Here, first character, which is that? Which is that? First character, round. That round allele, character for this one. Here, capital R. And the second allele, the recessive allele is coming from female gamete. The second female gamete, here small r. See this combination. Next, the capital Y, and here from the second gamete also capital Y. Okay, so here capital R, small r, capital Y, capital Y. Next combination, the first male gamete is going to combine with the third female gamete. Okay, here capital R, here also capital R. So both capital R came here. Next is capital Y and small y, capital Y, small y. Okay, so the third combination is capital R, capital Y, small y. And the fourth combination, here, the first male gamete, it is going to combine with the fourth female gamete, capital R. From here you can see the small r allele, then capital Y, then small r, y. See how we are going to write this combination? Here, it's very easy. Don't get confused. From this, you can get all the possible combinations very easily from this Punnett square. So how you will write? First character, both the alleles of first character from both the gametes you are going to take and write first. Then consider the second character, and from both gametes you are going to take the second allele. Okay, that's how you will form the combinations. Next we are moving to the second male gamete. See, this is the second male gamete. Second male gamete and all the possible combination with all these four female gametes. So second male gamete is having small r, capital Y. The gamete is having genotypic condition is small r and capital Y. So here, the first one is capital R from the female side and small r from the male side. Then capital Y from the female side and capital Y from the male side. Capital R, small r, capital Y, capital Y. And like that, the second combination also will come. In the second combination, what you can see? Here, small r and small r. Both are recessive alleles. And capital Y, capital Y. Other two are dominant. Our next character, both alleles are dominant. And same way, in the same way, small r and capital R. So capital R, small r you are going to write. Next is capital Y, small y. This small y and this capital Y. Okay. And the fourth combination, how it will come? Small r from this one, small r from this one. Then capital Y from this one and small y from the fourth female gamete. So the second combination also got over. Like that, you will get in total 16 different combinations. 4 gametes from male parent and 4 gametes from female parent you will get. So it will form 16 different combinations. Okay. Like that you can see 16 different combinations. So without confusion, you should write this. So while checking the result, Mendel got 3/4 of this showed yellow seed. Okay, 3/4 showed yellow seed. Here you can see. And 1/4 they showed green seed. Okay. So out of this combination, 3/4 Here you can see, you can count yellow color seed you can see here, and only 4. 4 showed in this Punnett square. I'm saying 4 showed which character? Green color, right? Green. And remaining 12, they showed yellow. Right? Count here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. All these 12 showed yellow color. And 4, here you can see 1, 2, 3, 4. 4 showed green color seed. Okay. So the yellow and green color, they segregated in 3:1. 12:4. That means 3:1. Okay. So this is in 3:1 ratio. Which one? Yellow color and green color. Yellow seed and green seed. In 3:1 condition. Right? And here again the round and wrinkled. Here you can see round and wrinkled. Round and wrinkled character also. Three is to 1, right? Here round yellow you can see, here wrinkled yellow. So both, that also again 9:3 means again it will form 3:1 condition. See these, all these combinations we are saying, see which here I am showing capital R, small r, capital Y, small y. What is the phenotypic expression of this particular combination? See, whenever some people are asking, whenever whomever is asking, you should be able to tell this phenotypic expression of that particular character. So this forms, how it will come here, dominant allele is there. So you can say it is round, right? And here expression of this dominant allele for the second character, yellow. So this seed will be round yellow. When another one, randomly you select one. Here the small one. Here this will be wrinkled because the recessive allele is present. This is recessive, and here again for yellow color dominant allele is present. So this is wrinkled yellow. Okay. So like that you should be able to tell the phenotypic expression or the expressed character by looking at the genotype. You should be able to tell the phenotypic expression. Okay. So you should practice with different combinations. You can try with different characters and you can practice writing this Punnett square and how you are explaining that characters. You should practice. Then only you can solve the problems when it is coming for NEET exam. Okay. So here in a dihybrid cross, the phenotypic ratio means the expression, character expression, that ratio is 9:3:3:1. Okay. The phenotypic expression, that phenotypic ratio of a dihybrid cross is 9:3:3:1. This you should learn, this ratio. In each cross, what is the ratio? You should learn that. And among this phenotypic ratio, round yellow, you can see 9 round yellow. From the expression 9 round yellow. Okay. Here, which are the 9 round yellow? 1, 2, 3, 4, 5, 6, 7. These and all. Here you can see round, the capital R, and yellow for dominant yellow color gene. You can see dominant allele, capital Y here. And here, next two. Here 1, 2, 3, 4, 5, 6, 7. You can see. I said 9. So here again you can see capital R and capital Y and capital R and capital Y. Round yellow condition. So 9 forms round yellow. Next, round green. Round green you can see. Where is that round green? Round. For round, which one you should get? Here capital R should be there. Then recessive allele. This capital R means round. In this also capital R is there. In this is also capital R. So round. Then you should get green. Green means it is a recessive character, so it should express in recessive, homozygous recessive condition. So here YY, small YY, here also small YY. So this will form round green. Next is wrinkled green. Wrinkled green. Wrinkled means it is the recessive allele of round form, right? So recessive expression means it should be in homozygous way. So smaller, smaller, wherever you can see smaller, 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 that is the recessive condition for the wrinkled condition. Then wrinkled yellow. Yellow means one dominant allele should be present. Yellow is the dominant character. So smaller, smaller, capital Y is present here for dominant condition and this also. So wrinkled yellow, three forms wrinkled yellow, this one and this one. Next is wrinkled green. Wrinkled green, both are recessive character. Wrinkled is a recessive character. Then again green, that is also another recessive trait or recessive character. So both are in recessive condition. Here only one combination you can see like that. Small r, then small y. That forms the wrinkled green color. So the ratio is 9:3:3:1. Okay, 9:3:3:1. That is the phenotypic ratio of F2 in a dihybrid cross. So next, here comes the phenotypic expression. Just we have already counted. Here this color shows 9 round yellow. Okay. Next, these three, those three shows round green color seeds. Next, wrinkled yellow. Here you can see wrinkled yellow. 1, 2, 3. These are wrinkled yellow. Next is wrinkled green. Wrinkled green. Which one? The recessive one. Wrinkled green. Okay. So how it will be? See, randomly we are selecting anything. Suppose we are selecting this one. How you will write the phenotypic expression? Here capital R is present. So it's round. Then small y. That is for color. How you will write? Round. It is in recessive condition. So you have to write the trait green. Round green. See, we are selecting this one. How we will write the phenotypic expression? Here capital R is present, the dominant allele. So definitely it will be round. Then Y, both are in homozygous dominant condition. So definitely it will be yellow color. So round yellow. See, next we are going to select this one. How we will write? Smaller shows the recessive condition. That means it's wrinkled. And next character. Here it shows capital Y, small y. Capital Y, the dominant allele is present. So it is wrinkled yellow. That's how we are going to predict the phenotypic expression by seeing this Punnett square. From the Punnett square, all the gametes, possible gametes and all the possible combinations you can make out very easily without any confusion. And by seeing that genotype, you can make out the phenotypic expression also. Got it? So the phenotypic ratio is 9:3:3:1 in F2 of a dihybrid cross. Okay. Next here, the law of independent assortment. Okay, by using this, based on the result obtained in a dihybrid cross, Mendel proposed a new set of second generalization. He made a second set of generalized rules and regulations. So these are called as Mendel's law of independent assortment. Mendel's law of independent assortment. This is the law of independent assortment. Got it? So this law, according to this law states that when two pairs of traits, they are combined in a hybrid, two pairs, we have selected two pairs for crossing, right? So two pairs of traits are combined in a hybrid. The segregation of one pair of trait. One pair of trait will segregate independently from the other trait. So there is no effect of the other trait for the segregation of this particular trait. Here two traits we are considering 1 and 2. This both characters 1 and 2 will be independently segregated. This will get segregated independently. There won't be any effect of one, segregation of one. This 2 won't affect the segregation of 1. And in the similar way, the second one, this character is segregating means there won't be any effect of this first character. So they will segregate independently. Okay. So with the help of this Punnett square which is given, now we will check with this help, with the help of this Punnett square, we will see the segregation of these characters. Okay. So if F1 plant, if it is a dihybrid, how it will be? Dihybrid means two characters. We have already told in F1 capital R, capital Y, small y. This is a hybrid condition, right? It is not in homozygous, it is in a heterozygous condition. And when it is selfing, how you will get? Capital R, capital Y, small y. This we are crossing with because it's selfing. This is selfing. Okay. So here the possible gametes which we will get capital R, capital Y. Another gamete is capital R, small y, then small r, capital Y, then small r, small y. So these are the possible combinations. And here also in the same way, capital R, capital Y, capital R, small y, small r, capital Y, and small r, small y. These are the possible gametes from this, right? So we can see here three different genetic conditions for this gametes, right? So in Punnett square, if we consider the segregation of one pair of factor in capital R and small r, this is first pair of trait, right? So 50% of the gametes will get dominant allele. Here you can see 50%. Out of this 4, 2 got this dominant allele for this character. And another 2 got the recessive allele of that seed shape. Right? So in the similar way, you can see here. In the seed color also you can see the segregation. How it is getting segregated. 50% will get the dominant, sorry. This is wrong. Here 50% will get how it will come? Capital R, capital Y, and capital R, small y. The small r, capital Y, and small r, small y. Now it's correct. So here 50% will get the dominant allele and 50% will get the recessive allele. Right? Again, in the same way, for the next character also. See here for next character, the seed color. 50% will get the dominant allele and another 50% will get the recessive allele, right? So there is a, here we can see the segregation of 50% of capital R or small r. It is in the independent way or independent, that is independent from the segregation of the seed color, this allele, Y allele, right? So similarly, segregation of Y allele, that will not depend, or that is independent from the segregation of the round and wrinkled alleles. So now we can see 4 types of gametes which are produced here, right? So each have 25% of frequency of combination, right? You got what I'm saying? Here, the allele will come in 50-50, right? First, 2 gametes will be having dominant allele. That means 50% will get the dominant allele and 50% will get the recessive allele of one character. Okay. And next, each here we can see 4 types of gametes in both sides of this Punnett square. And each one is having 25% of chance to combine with the other one, right? 25% of the frequency of combination, right? All 4 they are combining with, or one male gamete from this parent, that male gamete is combining with another 4 female gametes. In a similar way, all 16 type of F2 combinations you can see. So in the Punnett square, it is easy to derive this F2 combination. Just now already I have explained how to write these combinations. So the genotypic data, you need to find out the genotypic data. We have already told the phenotypic ratio, 9:3:3:1, right? So how to find out the genotypic data? Here genotypic data, that also I told from a combination how to write the genotypic condition. Here capital R, capital R, capital Y, capital Y. That is pure and pure rounded yellow, right? Both are dominant alleles that are in homozygous condition. That means pure round. Okay. Here one. I'll write all the genotypic ratio of everything. Of this F2, I will write the different combinations so it won't be confusing. The genotypic data of dihybrid cross genotypic ratio of dihybrid condition. Here you can see capital R, capital R, capital Y, capital Y. Okay. This only one you can see here. These are pure, round and pure yellow. Right? You can see only one. The first one you can see. This is pure, round and pure yellow. Next, another type you can see. In this you can see 2 capital R and capital Y, small y. Like this you can see 2 combinations. Okay. This is the one. You can see like this you can see. Another one also. These are pure round. Again, pure round, but this yellow is in hybrid condition, right? Hybrid yellow, right? This pure round shape is in pure form, but the yellow seed color, that is in a hybrid form. Next, you can see one combination like capital R, capital R. From this I am writing all combinations, capital R, then small y, small y. What is this? This is also pure round. But next one, next character is in recessive form, green. This forms green seeds. Okay. And another combination, again, 2 you can see capital R, small r. capital Y, capital Y. This is hybrid round, right? You can see different alleles for the shape, hybrid round. Because of the dominant allele, this also shows the round shape, but it is in hybrid form. Then YY, this is in homozygous condition. So pure which color it will form? Pure yellow. Yellow. Next combination is 4 genotypes. You can see capital R, small r, then capital Y, small y. This is hybrid round, hybrid round, and hybrid yellow. Okay, hybrid round and hybrid yellow. Next is 2. Another 2, you can see capital R, small y, small y. This forms hybrid round and green. Hybrid round and green. Okay. Then another one, you can see small r, capital Y. This forms wrinkled. Here this is recessive. So wrinkled and wrinkled plus yellow. Yellow pure, right? Here pure yellow. Next combination you can see 2 small r, capital Y, small y. Okay. This is wrinkled hybrid yellow. Right? Here yellow color, that is in hybrid form, but here homozygous recessive, so wrinkled. Next, last condition is this last one. Small r, small y. This is wrinkled and green. Okay. So this is the genotypic condition of all these combinations. We are selecting and writing all this. So here total you can see 1 plus 2, 3, 4, 6, another 6, 12, then 13, 14, 15, and last one 16. 16 different combinations we made here. From that we are making it according to the genotypic expression. From the genotypic and phenotypic, here we are writing the genotypic combination and writing the phenotypic expression also. Okay, so these are the different combinations. So how you will write the phenotypic ratio? 9:3:3:1. And how to write the genotypic ratio? Genotypic ratio in the sense the genotypic combination. How you will write? In monohybrid cross you wrote 1:2:1. Here how you will write? How you will write the genotypic combination? 1:2, 2:1, 2:4, 1:2:1, 2:4:2, 1:2:1. Okay, 1:2:1, then 2:4:2, then 1:2:1. This is the genotypic ratio. Genotypic. Here you can see in this ratio you can see different genotypic combination in F2 of a dihybrid cross. So This explains the law of independent assortment, that characters will assort independently. There is no effect of one character segregation with the another one. So the characters are independently getting assorted. Okay. So that's all about the law of independent assortment. So we have learned with the help of Punnett square how to write the combinations and how to write the genotypic expression and how to explain the phenotypic expression and the monohybrid ratio, genotypic and phenotypic. Now we have learned the phenotypic ratio and genotypic ratio of a dihybrid cross also. Okay. So that's all. So next comes a problem. Here after explaining this, we'll wind up. So in this problem, there's a question in Pisum sativum or Pisum, the yellow seed color, why it is dominant over green color. Okay. Yellow color, it is dominant over green color. And round shape. The round shape of seed R, that is dominant over wrinkled. This is yellow and this is green. And round shape and wrinkled shape. These are the alleles for that. So here, consider that these two pair of gene assort independently. There is an independent assortment. Then what proportion of the offsprings from the cross? The cross? Which cross they have mentioned in the first question? They have mentioned the cross capital Y, small y, capital R, small r. A cross with small y, small r, small r. This kind of questions you can expect for your NEET exam and for your board exam also. So here these are the parents. So here the first parent, how it will produce gametes? It will produce, these are in heterozygous condition, these characters. So it will produce 4 types of gamete. Capital Y, capital R, that is one gamete. Next, capital Y, small r, that is another second gamete. Then small y with capital R, small y with small r. This parent. How about this parent? That will produce only, both characters are in homozygous condition. So that will produce only one type of gamete, right? Small y, small r. Then we can write all the possible combinations. When capital R, capital Y, capital R and small y, small r. This is combining means it will give capital Y, capital R, small r. Then next, this combination. How it will come? Capital Y, small r, small r. Then next combination. How you will write? Small y, capital R, small r. And the last combination you can see small y, small r, small r. So what is the question here? What proportion of the offsprings from the cross? This cross would expect that to have yellow seed trait. Yellow color, we said yellow color, it is the dominant color. So here, yellow color seed trait, how many of these two are the recessive alleles? So it won't produce yellow color. This will form green color, right? Here, these two are having allele for yellow color seed. So here, out of this 4, 50% will form or it will have seed color yellow. Okay, that's how you're going to solve the problems from this chapter. Got it? Then, next question from the cross, capital Y, small r, small r, capital Y, small r, small r. How many will be pure yellow wrinkled plants in the resulting genotype? So here, how many types of gametes it will produce? Capital Y, small r and small y, small r. These are the two possible gametes, right? In both cases, capital Y, small r and small y, small r. These are the two possible gametes. And different combinations you can see. These two. Capital Y, small r, small r. Next, these two. Capital Y, small r, small r. Next, this one and this one. Capital Y, small r, small r. And last, these two will combine and it will form small y, small r. Right? So these are the 4 different combinations in F2 generation. So here, the question is yellow ringled. So to get yellow ringled, which one will form yellow ringled? Yellow and wrinkled. How it will be that condition? Pure yellow wrinkled they ask. Pure yellow wrinkled. Here yellow wrinkled means this also will form yellow, this also will form yellow. Okay, these both will give wrinkled. But pure they have asked. Pure means only one. This capital Y, pure yellow and pure wrinkled. This combination. So only 25% of the plants will form yellow wrinkled, pure yellow wrinkled condition. Got it? So that's how to solve the problem. Hope everything is clear. Do you have any doubt? Is it clear?
No. Yes, ma'am.
This problem is clear?
Yes, ma'am.
Okay. So we'll see in the next class.
Thank you, ma'am.
Thank you.